future scientists struggling with endospore staining or confused about malachite green and heat fixation, worry no more. In this 10 in this 10 minutes crash course, we will decode endospore staining right from theory to practical tips, even with some Viva hacks that are going to help you a lot during your practical exams. I'm Dr. Priyanka, and you are watching Cosmic EduTech, your smart companion in life science. So let's dive in. Let's first understand what is an endospore. Endospores are tough, dormant structures formed by certain bacteria like Clostridium and Bacillus. So, especially they occur during hard conditions. So, we can think of it like a bacterial survival suit for the bacteria. They resist heat, chemicals, UV radiation, but there is a catch here. Ordinary stains like crystal violet, saffronin, they cannot penetrate them. So we need a special technique that's called endospore staining. The objective is to differentiate between spore forming and non-spore forming bacteria and highlight endoscope within the bacterial cell. Think of it like you want to dye or stain a coconut. So you'll need to crack open it with something, okay? But here, in case of our staining, we are going to use heat before we open it, right? So you will have to crack it open with heat before the color gets in. Use a method called a schaeffer Fulton method, which uses chemicals like malachite green and saffronin. So the process is such that malachite green is going to act like a primary stain and which will be taken up by the endospores only. Heat is going to allow to force that stain into the tough spores. And finally comes the secondary stain, that is your saffronin, which will be taken up by the vegetative cells. So that's why we have the endospores which will appear as green in color, and the remaining cell, the vegetative cell, will look like red or pink. So heat helps malachite green to enter inside the spore coat. So without heat, stain is not going to bind it and you will end up having a colorless cell. Go through the protocol. So here is a perfect practical protocol that you need to follow to get perfect endospore staining. So what are the requirements that we need here? Very first one is a bacterial smear. So mind you, your smearing technique should be perfect and you have to use a fresh culture, that is a 24-hour old culture. So ideally for practical purpose we do use Bacillus subtilis. Then malachite green, which should be fresh again. We should not use from the ancient bottle from the ancient racks. So it is 5% aqueous. Saffronin, that is going to be your counter stain. Then burners, lights, okay, racks, okay, staining racks and distilled water. So let's move on to the procedure that we need. The procedure seems to be very simple, but here are few things and tricks that you need to be very perfect with. So prepare a heat fix smear on the slide. So the slide should be perfectly and nicely clean. It should be grease free. Then your smear should be even and very perfect. Place the slide on steam bath or a beaker with boiling water. So after that, cover the smear with malachite green. So make sure it is on steam for almost 5 minutes. So in case if you find the stain has dried up, make sure you are adding some few drops of fresh stain on that. And after that, cool the slide and keep it aside. Rinse it gently with water and pour the counter stain, that is saffronin, keep it for 30 seconds of contact. After that, rinse it, blot dry and observe it under oil immersion microscope. Under the microscope, you will get some image like this that I have put up here. So you will find green ovals which are going to be your endospores and the pink rods will be the vegetative cells. So steam is the key. Without it, spores will remain colorless. So if you do not use the steam properly and if your malachite green is not proper, you are not going to get a good stain. Let's see what are the do's and the don'ts that we have to follow in endospore staining. So let's see what are the common mistakes that we can avoid. So first let us consider the do's. Always use a fresh culture, so 24-hour old culture. Maintain moist heat, so we should not allow the stain to dry, and cool before rinsing. These are the things that you need to keep in mind. Next, what are the don'ts that we have to follow? Do not overheat the slide, that is going to kill the cells. Next, don't skip the counter stain. So, if you forget the counter stain, you will end up having a big green blob. Next is your malachite green should be new. It should not be old because it is going to give you very poor results and your staining will be not so good. Please remember clarity. Your marks in the practical exams will depend on the clarity of the slide. Always call the examiner when you have chosen the best field that you can present to your examiner. All looks blank or everything is green, so don't panic. So I'm giving you some of the tips and this troubleshoot guide over here, which will help you definitely to understand your mistakes and solve them immediately. Suppose there are no spores visible, so probably you have used old culture or you have not applied heat properly. So rinse it off, use fresh culture and use proper steam. Next, if the whole slide has turned green, that means there are two things possible. One is either you have overstained it or you have forgotten to use saffronin, that's your counter stain. So again, rinse it, add saffronin. Third point, if the slide has dried while steaming, so don't, it is not going to keep your stain moist. So add fresh stains, right? And finally, if your spores look a bit faded even after staining, that means you have definitely used the ancient malachite green. So wash your slide and use again the fresh malachite green. So these are some of the things that you need to keep in mind that is going to guide you understand what mistake you have done. Before we actually wind up, let me give you some easy tricks. So let's log this in your brain with some funny mnemonics. So green spores survive, red cells die. So this is the thing that you have to remember here. So green is for the spores, red is for the vegetative cell, and survive, that means so spores survive in harsh conditions. So we can have a jingle like, spores are green, they don't scream. They are in the survival mode extreme. So this is something that we can remember this endospore staining. So before we formally end this, so let me give you some of the Viva tricks or Viva questions that could be asked for endospore staining. Like for example, what is the use of heat in endospore staining? So here the point we have to remember is that malachite it is needed for malachite green to penetrate inside the spore wall because the spores are very tough. Then next is why are this spores green and the remaining cell is red? That is because the endospores are going to retain malachite green whereas the cell is going to take up saffronin. So another thing is why can't we use gram staining because it is a very well established protocol, right? So the point is because of the tough nature of the spores, your gram stains do not enter. So we will not get a good staining. Please remember, malachite green is water soluble and it does not bind strongly. So it needs heat. So this is one thing that you have to remember. And that's it. You have mastered the endospore staining right from theory to Viva and you are now exam ready and lab smart. So like this video, share this with your batchmates. Comment below if you have any doubt. I will definitely try to solve your doubts. And subscribe to Cosmic EduTech by Dr. Priyanka for more such life science hacks that actually make sense. So till then, keep studying and keep smiling.